Today, our speaker, Dr. Allyson Muotri, is a man of many brains. Not just his own brilliant brain, but the many brains that he has created. And today, he has a major challenge. And his major challenge, probably even harder than figuring out how to send the many brains into space, is how to help us understand the complicated research that he does and get that understanding into our brains. Like, for example, what the heck is a pluripotent stem cell? I hope you'll feel free to ask him questions when you don't understand, and he will take the questions after and give you more time if you're shy about asking in public. And here to introduce him properly is Payleen Thorogood, who spoke to us a few months ago about CBD research, and she's the president and founder of Holistic, a philanthropic group that has funded Dr. Muotri's studies. So, Payleen.
Thank you so much, Rena. Hi guys, it's so nice to be here and see so many faces. I am so honored and delighted to introduce my good friend and incredibly brilliant researcher, Dr. Allyson Muotri, who is a professor of pediatrics, a professor of cellular and molecular medicine at UCSD, as well as the director of the UCSD stem cell program. I met Allyson about 2 years ago when our foundation, Holistic Research and Education Foundation, was looking to fund CBD efficacy research with UCSD. They came to us with a very interesting study around studying how CBD may help with autism. It was a clinical study. And while it looked interesting, we thought it was not going to provide us enough information on how and why CBD may actually benefit autistic kids. And at that point, I met Dr. Muotri and also another brilliant scientist, Dr. Gabe Silva, who augmented the clinical trial by adding the ability for us to collect a lot more data than just a few kids that were going to be in the clinical trial by leveraging this mini brain concept that he's going to talk about today. To me, his work, which of course spans a lot more than what we're doing with him, provides us an ability to collect significantly more data, be able to do far more experimentation using human-derived organoids without actually having to give the medicine or the treatments to actual humans, but still see its effects as something close to a human brain versus working with animal models. So it gives us a lot more data, provides us an ability to actually understand the mechanisms of action. It gives us an ability to understand the how and why behind why certain treatments might be beneficial for specific conditions. So to me, it's really the future of science. It really enables us to get into human models a lot more easily without actually impact to individual humans. So with that, let me read a little bit about his background with glasses on. Dr. Muotri earned a PhD in genetics in 2001 from University of Sao Paulo in Brazil, and as you'll note, he's Brazilian. He moved to the Salk Institute as Pew Fellow in 2002 as a postdoc. And since 2008, he's a professor at UCSD in the departments of pediatrics and cellular and molecular medicine, as well as director of the stem cell program. His research focuses on human neurodevelopment, modeling neurological diseases, and brain evolution using cerebral organoids. And he has received several awards, including the prestigious NIH Director's New Innovator, NARSAD Rockstar of Innovation, and NIH Eureka, among many others. I expect many more to follow. So, without further ado, Allyson. If you guys could all turn off your cell phones just in case. Thanks.
Thanks so much, Payleen. That's a very kind introduction. And thank you all for the invitation. And thanks for coming even with the coronavirus around. Yeah, so my lab focus on this idea of modeling the human brain, and it's a major problem. And the motivation to do that is because neurological and neurodevelopmental disorders affect many people. So this is my son, Ivan. He has autism and epilepsy. So it's very, very hard for him to deal with even normal interactions, normal communication in his daily life. So he struggles with many seizures per day, so that's not easy. And the interesting part is that we don't know what to do. I mean, doctors don't fully understand the human brain to come up with treatments for these kind of conditions. Most of the treatments that we have so far was discovered by chance, just trial and error. Very few drugs actually have a very clear mechanism on how it acts in the brain. The later blockbuster drug for the brain is Prozac from the 70s. So since then, there's nothing. So we keep like rebranding the same old drugs and selling them with different names. That's what most of the time that's what happens. It's a different situation from cancer or heart disease. You might see advances. There are some forms of cancers that we can already cure, and nobody dies for heart disease anymore. Very rare for this to happen, especially if you get up early on. So why the brain is so difficult? So here's the reason why it's so difficult. Because we don't fully understand how it's formed. And I'll give you like a very clear example. This is a car. Everybody sees a car here. Because we recognize all the small pieces, we know how the pieces should look like when the car is assembled. So the challenge that I make to my colleagues, neuroscientists, is that if I break out the brain in different pieces, nobody will recognize that is the brain. Because we don't recognize the pieces, we don't know how it should be put together. And you might ask, come on, I mean, there is neuroscience research going over for several years, how come we don't fully understand the brain? And here is the catch. Most of neurosciences is focused in the adult brain. So it's like me looking at my computer and being enthusiastic about its complexity, but I don't know how the computer works. And the other part of neuroscientists, they look into a mouse brain. And a mouse brain follows very different evolutionary trajectories than the human brain, so it's not the same. So even though you might understand how the mouse brain works, you will never understand how the human brain works. So, and the reason why we don't study the human brain while it's developing, it's because it happens in uterus. So the human brain is formed inside the uterus, so we don't have ways to study it when it was being formed. So we have to rely on technologies such as an ultrasound. That's a very indirect way. By the way, there are 2 pixels in there that represents the brain. You cannot see a single neuron. You cannot see how these neurons connect to each other. So we don't understand how the brain is formed by just looking to ultrasounds and other methodologies. So then, I mean, we have to come out with models and this idea of what would be the best model for the human brain has been around for several years. We all try to have the better models. But we need first to understand what is a model. And it's so funny because when I ask for different people, I get different responses. So I'm from Brazil, right? So when I ask my colleagues what's a model, they said, this is a model. And then I think and said, oh gosh, they are right, that's a model. So if I ask a kid, they will say this is a model of a train. And if I ask an engineer, it's a model of a flying machine. So they are all right. And models are simplified representations of the reality. The best models are the ones that get closer to the reality by adding complexity. So what are the models we have so far? So we have post-mortem tissue. These are people who donate their brains after they're dead. But here's the deal. I mean, these post-mortem tissues are not in good conditions. They take several hours for them to remove from the body and go to the scientists. The cells are dead. I mean, those neurons are not active, so we cannot study. Most importantly, the brain is already fully formed. And I want to understand what happens before the disease appears. So it has several limitations. Other things that we use is gene expression or gene analysis coming from peripheral tissues such as blood. But blood is not brain cells. So again, we don't have access to brain cells or how it communicates. And I mentioned about mouse models. Even monkey models do not represent even close what happens in the human brain. So we are very limited with those models, so there's nothing we can do. And that's where stem cells come in place. We can use human pluripotent stem cells. These are human stem cells that are close to or similar to the embryonic stem cells that can give rise to several tissues in the body. And we can teach these cells how to create what we call, AKA mini brain a dish. And you see this name mini brain, it's not like the most accurate brain because it gives you the impression, accurate term, because it gives you the impression that you have a fully formed brain in a dish, and that's not the case. I'll show you exactly how it looks like. And the correct scientific term is a brain organoid, or a small organ of the tissue. So these brains in a dish or organoids also have limitations, and I'll just lay down some of the limitations here. Most neurons are immature. These are tissues that are not vascularized. We don't have all cell types. We don't even know if we are growing them in the right conditions because we don't know how the human brain grows. So these are some kind of challenges that we scientists face in our experiments. But in any case, I mean, there are ways to create human brain tissue from these stem cells. And this is a recipe that took us about 4 years to develop. And we start from those human pluripotent stem cells, and through a series of different media formulations that we add to those cells, we can make them self-assemble and proliferate, grows, and then mature into a fully formed, not a fully formed, but a piece of a mature cortical tissue. And we can do that for different brain regions as well. So the interesting part about that is that those pluripotent stem cells, they don't need to come from the embryo. We can take them from any live person. So if I take a piece of your skin or your hair cells or your dental pulp, I take it to my lab and I reprogram these cells back to this pluripotent state, and that cell now erase all the memory, all the identity, and they become naive stem cells. So from that stage, then I can take all the way and create brain cells. So here's an interesting point. If I take a cell from you, I capture your entire genome, your entire genetic information on that pluripotent stem cells. When I make a brain organoid from that cells, it's your brain organoid because it contains your genome. So it's an avatar of your developing brain in a dish inside the lab. So it sounds like science fiction, but it's not, because we have many of them growing in the lab. This is a postdoc in the lab. She's handling like a plate, and you're going to see some white dots inside these wells. Each one of them is a brain organoid. So when we make them, we don't make one or two. We make thousands of them for every individual that we study. So right now in my lab, we have 30,000 brain organoids from different individuals growing at certain stage. So the beauty of that is that you can use different of these brain organoids to do different experiments at the same time. And, but let me tell you a little bit how they are formed. I told you that they self-assemble. So we don't have to tell them or to tell each cell where to go. So the cells, they actually migrate following some of these cables. I hope you can see there are some cables. These are called radioglia cells, and they guide the formation of the human cortex from inside. This will be the center of the organoid, inside out. As they migrate out, they stop and they start forming connections with each other. This will be giving rise to the cortical plate. So if I section one of these organoids in half, what you see is a structure like that. There is a ventricular zone. These are the neural stem cells. These are the radioglia cells showing the process here. And these are the cells that are migrating out, forming the cortical plate. So the anatomy is quite striking. It's even a miracle that we can do that. If you asked me 10 years ago, I would say that would be impossible. We would have to learn how to put each cell in the exactly location. But what we learn is that they are genetically pre-programmed to be in the right place at the right time, which is phenomenal. It saves us a lot of time. And this is a way to show you what are the different cell types that we have inside one of those brain organoids. So each dot is it's a single cell, and the colors shows the different identity of these cells. So you can see there is a population of progenitor cells here. These progenitor cells will become intermediated progenitor cells, and they will fully differentiate into glutamatergic neurons. These are neurons that produce information. They're excitatory neurons. These are glia cells. They keep the neurons healthy. They also monitor your brain status. These are the inhibitory neurons. This population will eventually grow more over time, so it becomes more balanced. So in the beginning of neurodevelopment, it's all about excitation. There's lots of inputs coming. But as we grow older, we put it down. So that's a sign of maturity. And we see that being recapitulated in our organoids. All right, so we have the cell types. We have the anatomy. So as soon as we start seeing those things, I mean, we start using to model some of the diseases that affect brain formation, because that's exactly a very nice system for that. And I'll give you like a striking example of how this technology was able to solve a very important problem. Coming from Brazil, I knew about the Zika virus way before it became an outbreak. My colleagues were saying, oh, we have kids born with microcephaly, and we think this has two potential causes. It could be a virus that nobody heard about it, called Zika virus, or it could be the pesticides. So we don't know. How would you test that? So I got an aliquot of those Zika virus, and I put into a mouse model, and it was a female animal, pregnant, and all the babies were born normally. So my conclusion was, well, the Zika virus did nothing to these animals. But then, in parallel, I add the Zika virus to one of these human brain organoids, and after 2 days, it was gone. All the cells were dead. Yeah. So why? Why did not show anything in the mouse models? And the answer is developmental gestation time. I can get a fully formed mouse brain in 20 days, but takes 9 months to have a baby's brain. So the human cells are very slow dividing. They migrate very slow, while the mouse cells, they do it really fast. So we couldn't see the effect with a very small amount of virus in the mouse models, and someone that does not look for the human model would completely miss this opportunity. And once we established that there is a causation between the Zika virus and microcephaly, we could see if there are any way to repurpose antiretrovirus to block the action of the Zika virus. And in 2 years after the outbreak, we found sofosbuvir, a drug that's already FDA approved for hepatitis that could be repurposed to block the Zika virus in the brain. So now, in case of a new outbreak, this drug will inhibit viral replication and could even inhibit what we call vertical transmission. If mom is infected, we can protect the baby's brain if she takes this drug. So it's one of these examples where the model was in the right time, so we could save, we could solve a very complex problem that would be very difficult to solve if we didn't have these kind of models. So with this idea, we start to ask, wow, I mean, this is a powerful model. Maybe we can understand everything that affects the brain in uterus. And so some of my colleagues think that the uterus is just an incubator, but it's actually a protective environment that dictates how the brain is formed and who you're going to be later on in life. So we should not neglect what happens in uterus. And there are many things that you receive, many chemicals from the environment that you receive when you are pregnant. And what we wanted to do is to create a way to pre-screen all these chemicals before they cause any harm. You probably heard about thalidomide. Thalidomide was approved. It passed the animal toxicity test. We didn't have brain organoids at the time. You probably heard about Tylenol being associated with high chances of autism, ADHD. So Tylenol, why we are only catching these now? Well, because epidemiology takes time to reach out. So what if we can give them like a seal of approval, and everything that a pregnant woman will take will have to pass to this kind of toxicity test? So at least, it would be very hard to say that it's fully safe, but at least you can red flag if something creates a problem. So that's a way to improve our toxicology treatments. So another thing that happened in the last 2 years was once we start solving all these problems with the Zika virus, I got lots of reporters calling me to talk about this model. And there is one question that everybody was asking. So you have mini brains in your lab. Do they think? Sounds naive, right? But made me think, yeah, that's a great point. And we tested. And most of the protocols we had at the time didn't have neurons that were active. So they never formed an active network. So we went back to the lab and we optimized the conditions to grow mini brains with active neurons. And so this is one of the experiments that we did. You can place one of those organoids on top of a multi-electrode array. So these are electrodes that are printed in the bottom of the dish, and you just plate the organoid on top, and you record all the time when there is going to be an activity passing by. You're going to generate a heat map like this one, showing that those electrodes become active. Over time, you can just record, and you see that every electrode will produce what we call a raster plot. So every trace, it's a spike, is when there's an activity in there. So in the beginning, I was not optimistic that this approach will work. And the main reason why, it is most of the studies before, they could never pass what we call the 5 Hertz threshold. So if this is just a brain activity, and this is time, most of the studies are here below 5 Hertz. And a primate brain, it's about 20 Hertz, and a mouse brain is about 18 Hertz. And our human cells in a dish is below 5 Hertz. Again, we could never make this to work. But I show you now the result that one of my postdocs presented to me after he optimized the conditions to grow them. So this is what we got. So over time, we see that the activity starts to increase, and by 40 weeks, or about 9 months, we are reaching this 20 Hertz threshold. So 20 Hertz threshold means tons of activity. Those neurons are firing like crazy. They are really producing activity. And if they are that active, they also produce what we call brain neural oscillations. So these neural oscillations, or brain waves, is what you can read from an EEG, an electroencephalogram. When you place those electrodes through your skull, the brain has so much activity, you can read through it, and these are the waves that you can record. These waves are very important because it tells you different behaviors that you might have. For example, if you have a seizure, these brain neural oscillations will change dramatically. If you have schizophrenia, they will change. If you have autism, they will become really slow. If you are paying attention, there is certain shapes of waves. If you are sleeping, it's a different wave. So it tells you how you feel, what you're doing. So that's why it's very important to measure that. So we team up with Brad Wojtek. He's from Cognitive Neurosciences at UCSD. And we gave him the data, and he started measuring these oscillations using his methods. And what he sees is that these oscillations start about 4 months. When they reach 6 months, they're very stereotyped, very synchronized. And if you wait longer enough, after 8 months, they become highly complex. too complex. We've never seen this before. It's a level of a complexity that it was unprecedented. So we start asking, does this match what we see in the human brain? So I cannot place electrodes in a baby's brain in uterus, but I can place electrodes if I have a baby that is preterm, premature babies. So when we do that, we see that the baby's brain, they have a very specific way to become active. That's what we call trace discontinue. They have peaks of activity followed by a quiescent or silenced time. We always started this way, having activities, silent activity. As we grow older, the silent period in our brains becomes shorter and shorter and shorter and shorter, so give rise to a very complex EEG like this one. This is what we all have, a very complex EEGs. So our brains will never, our brains will only return to this state in one condition: minutes after death. It's funny, no? You go back to your embryonic stage before you die. Yeah. So, but look at the organoids. The organoids look like the premature brain. So, I mean, but how close is that? So what we did was to create a machine learning algorithm where we trained the machine to tell the age of the subjects based on preterm's EEG that spans from 25 to 38 weeks. And the machine does it really well. It looks at the EEG, tells the age of the person. And then we feed the machine with the data coming from the organoid, and we ask, can you predict the age of the organoid? And the data is striking. So the perfect correlation is in this bar here. The real data is in black. The organoid data is here in blue. You see that it follows the same trajectory. It's more variable. It's not perfect. But it follows the same trajectory, which is amazing. And before 25 weeks, the correlation is really bad. And the reason why, we have no data from humans' EEG before 25 weeks. Most of the babies will not survive. So we still don't know what happens in the first 25 weeks. But nonetheless, after 25 weeks, it seems like our model can recapitulate some aspects of the electrophysiology of the human brain, which is amazing. So now, if you are a UCSD professor and you recreate an artificial brain that fires like the preterm brain, what happens to you? You end up in the New York Times. So that was August last year when we published that. So they made an article because now we are opening different questions, including some ethical dilemma. How should I treat these brain organoids? I mean, are they self-aware? Are they conscious? Do they have a moral status? Do they belong to the person that they were derived from? I don't know. We are challenging even the philosophers who never thought about that. So yeah, it opens like some very interesting questions, but I'm more excited about the applications. What can I do with this model now? And I'll share some of the projects and ideas that is going on. I think Pei-Lin mentioned to this gift to run a clinical trial with cannabis, CBD, for autism. So these are 30 kids in a clinical trial, double-blinded control placebo, and we will follow them for a while and see if they improve or not in different aspects of autism, such as aggressivity, stereotype behavior, and many other features. But in parallel, what makes this so interesting is that every single of these kids will have their mini brains made by my lab. And we're going to be testing CBD on their mini brains as well, in parallel. So what I'm hoping to take out from this study, it is a signal that can tell me from the brain organoids, from these mini brains, what happens in the brains of those kids. So I can understand how CBD works. I can understand why some people respond to the drug and others not. I can understand what is the optimal dose for each one of the subjects. So that's the ultimate goal of this project. we also make an effort to recreate some neural circuitries. And one thing that we were very curious about it is that after 40 weeks, I show you that curve of activity, that it reaches these 20 hertz. And what happens after these 9 months, it is that it plateaus. I can keep these organoids alive for several years, but the activity plateaus. It doesn't continue to evolve. And we thought that, well, there's many reasons why this could be, but one thing might be that at 9 months, the brain is ready to receive inputs. So that's what happens to a baby. It opens its eyes and sees moms and start forming that connection. This visual system is really important in autism, but in many other conditions. So we thought maybe we could recreate a visual system by connecting a mini eye to a mini brain. So we create those three-way organoids. What you have here, it's a mini retina created from, again, embryonic stem cells. This is a mini thalamus, and this is the mini cortex, the mini brain. So the photoreceptors are labeled here in fluorescent green, and the projections of these photoreceptors are labeled here in red. This is a collaboration with Carl Walling here in the ophthalmology at UCSD. And we are hoping that these photoreceptors will mature the cortex, and we can stimulate with light and record from the cortex. So that might increase the way the networks mature, and we can push even far how these brain organoids represented the human brain. We also have like very basic questions, such as, can I store a memory in one of those brain organoids? That would be fascinating to see. So this is, we are trying here to recapitulate the biology from the eye to the brain. But I thought, what if we stimulate these mini brains using different systems? And instead of giving them just an eye, why don't we give them like an entire body, right? Maybe they will have like more stimulation. That's very hard to do. But we thought about a robotic interface. And so what you see here is a robot that is moving or is learning how to move and coordinate the legs using electrical information coming from one of these human brain organoids that we recreate in vitro in the lab. It's amazing, no? So the brain organoids are inside the incubator. We are recording from them, and we are sending the electrical activity wireless to this robot. We just add those two eyes. These are not eyes. These are infrared. These infrareds are detecting when the robot gets close to an obstacle. And all the time when it gets close to a wall, it stimulates back the organoid. The networks will now change. And the computer who makes the interpretation will see that the networks have changed and will send a second command to the robot, such as walk back. So if you come to my lab, you're all welcome to visit. You're going to see like a small robot trying to navigate like an environment. And by doing so, it stimulates back the organoid. So why we are doing this? So I told you, it's not to recapitulate human biology, because the organoid has no clue, no idea that's controlling a robot. We are more interested now on the stimulation that's coming from the robot. So we team up with Gabe Silva, the other investigator who does computational neuroscience, and we just got an incentive from Microsoft, because we want to recreate these networks and apply to artificial intelligence or design new algorithms that mimics the learning that the human brain does, and not an artificial intelligence algorithm like neural networks that we currently have. So this would be like a more human-like artificial intelligence, but coming from an organic brain organoid. So if it all goes well, in the future, all our cell phones and cars will navigate the world using this kind of network. So that's the goal.
Siri will get smarter, right?
Yeah. That's an improvement. So the other thing that I want to mention to you is this idea of learning how the human brain evolved to become human. And I have this project in the lab that looks for the evolution of the human brain, and we have been generating these organoids from different animals, so I can compare how their brains evolve. So we go to the San Diego Zoo and we collect cells, and we have like an entire brain zoo in the lab. And we compare them. And even a chimpanzee falls apart from the humans. We are outliers. We are so different from all other species. And that's why I'm upset with the animal models. So, but if I want to understand how the human brain evolves, I need to understand how other humans have evolved. And I'm not sure how many of you know, but we are not the only humans. There are many humans that have lived here on Earth, and we were just one of them. These are several fossil records. The ones that we understand the most are the Neanderthals, because we have lots of fossil records from the Neanderthals. And they have been here for a while, even longer than us. And the only thing that we can understand from them is based on fossil records such as endocast. We can extract DNA from their bones, and we have some impressions that some people might argue that this is abstract art or not. But there is one thing that I found it amazing about the Neanderthals that I want to share with you, is this idea of adaptation, art, and technology. From the fossil records, if you get old fossil records from the Neanderthals, you have stone tools as a technology. If you have more recent fossil records, you still have stone tools. They improved. You might don't know, but it's an improvement from one to the other one. But now when I compare to modern humans, we start with stone tools, but we are here having this conversation. So we accomplished so many arts, technology, and adaptation to really dominate as a species in the world. Why this happened? Why we are the only ones, only animals in the planet that can do that? So what I want to do is, you probably figure out, I want to create a brain organoid from a Neanderthal and study that in the lab. But there is a caveat here. There is no there is no live cells. The only thing we can get from these bones are DNA. And so I decided to take like a very different approach. Instead of a cellular approach, let's have a genomic approach. Let's get the DNA from the Neanderthals and compare to modern humans and ask, what is the difference between us and them? So we have about 20,000 genes, protein coding genes in the genome. And there is about 40, 60 genes that are different depending on who you ask. These are small alterations in your DNA that separate us from the Neanderthals. It's a small pool of genes, but they're very important because they control your immune system, your bones, your muscles. But a couple of them, actually 3 of them, actually control your brain development. So if it's only 3 of them, I can go back to my cells and use genome editing tools to insert back the Neanderthal version of those genes inside the cells. And then I can recreate these Neanderthalized organoids, or Neanderthoids as we call them in the lab. So the interesting part is that these are point mutations. These are single mutations in your DNA that make us different from the Neanderthals. But again, even a small number of genes might have a dramatic impact on how your brains form. And that's what I'm searching for. And we were lucky because we found something. These are how the modern human brain organoids look like, I told you. This is how the archaic brain organoids look like. You can see that the morphology is different. This might indicate different ways that those cells migrate out to form the cortex. But also fascinating is you plate them on these multi-electrode arrays and you record their activity. And you see that this archaic version of the genes produces networks that are way more active than us. So this is comparing to us. But more activity doesn't mean that it's better or worse. It just means that it's different. So similar to a chimpanzee who can mature really fast, the Neanderthals can mature really fast, but the level of synchronization that we see is really low. So again, we are seeing genetic evidence that our brains are way more sophisticated than even the Neanderthals by doing this genomic analysis. So we are here really for a fruit of a small pressure or a small random event during evolution that actually made us succeed in this world. So it's very interesting to think about autism and complexity of the brain. All these mental disorders are probably an evolutionary trade-off between complexity and brain function. All right, so if I can use this technology to look into our past, maybe I could use the same technology to look into our future. And I can start creating brain organoids that carries some advantage that we don't currently have. So these would be ways to manipulate the genome in our cells to create brain organoids that are very different, that evolution has never had a chance to test. But I can speed up evolution in a dish. For example, can I manipulate the genome to create a brain organoid that is resistant to dementia, resistant to Alzheimer's? Can I understand what makes a brain resistant to autism? Things like that. So, I mean, it opens another ethical dilemma, and we can discuss that later. But this is the kind of project that really push the science forward and make us think. And a similar project was the one that Palin mentioned, or I think Rina mentioned, about sending these organoids to the space station. So it happened last summer. In July, we sent organoids to the International Space Station so they can develop in the absence of microgravity. This was inspired by the NASA Twins project, where one of them stayed on Earth. The other one was spent like a year in the International Space Station. And what happens when he returned is that there's lots of systems that were affected, including cognitive decline or ocular problems. So when NASA sees this kind of data, it worries because, I mean, if they want to send a man to Mars, it will take more than a year. And what will be the problem and how to mitigate that problem from the astronaut health. So one way to do that is to recreate ways to grow brain organoids in space. So we use a cube like that. The brain organoids are in these tubes here. We have all the microfluidic chambers here that will feed these organoids while in there. So the astronaut doesn't have to do anything. So we go to the Cape Canaveral, got the seal of approval. Initially, when we started this project, I dedicated the project to Dennis Sanford because I thought that he was an amazing cheerleader for stem cells, and he was visionary as well. So I thought, well, I mean, this is the kind of project that we should honor Danny. And he was so kind that he flew over there to see the launching with me. So I was very happy to see. And so the first day, actually, it didn't go. And I said, oh my gosh, Danny's here. He traveled all the way. And because of the weather conditions, they couldn't fly. And so we have to stay one more day. But then, I mean, yeah, the next day was all good. And then, yeah, we all celebrate, have good time. And so the organoids finally reached the space station. Again, the only thing that the astronaut has to do is to combine these boxes with this board and plug inside one of these cabinets here. As soon as it's plugged, I have immediately access to all the sensors and cameras and everything inside that box. And I couldn't resist but take a picture. So this is inside one of the tubes. The camera needs to improve. We're working on that. But I hope you can appreciate that there are some spheres in here. So these are the organoids. They seem to grow a little bit faster than here on Earth. They also are perfectly round. The ones that we grow here on Earth, they are not perfect. They are spheres, but slightly different from each other. In microgravity, there's something about the cell migration that they are all very different. So we're very excited about that. And so one of the potential applications on Earth of this technology is to study neurodegeneration or diseases that has like a late onset, diseases of aging. And this is because when you bring them back, they age faster. So it's a model for, again, accelerated aging. These organoids, they will accumulate lots of DNA damage, metabolism. Their telomerases, which regulates aging or age of the cells, will also change. So now I can start studying diseases like Alzheimer's or Parkinson's or any other late onset condition if I grow them in the International Space Station and then bring them back so I can do that. So that's one way to apply that. So the other application would be to grow them, for example, this is a vision that we have in the lab. We're going to change the way we grow these organoids. We're calling them brain farms. So we're going to have all these boxes that will autonomously grow in these organoids in a much more optimized way. So we don't worry about variability. We don't worry about cost. So we can dramatically reduce the cost by growing them in that way. But most exciting, it is what I don't know I'm going to find by growing them in microgravity. So this is what we call a leap in biology. It's probably, microgravity is probably the next force. I mean, we experiment with microwave, we experiment with magnetic forces, but microgravity is inaccessible to us. The only way to study microgravity is to go in there. And so there's many things that we think we just don't know. So probably a better understanding or better models, even for autism, would be achieved by growing these organoids in space. So that's our hope. So I talked too much. I'll stop here to take comments and questions. But before that, I want to mention a very nice resource. As a director of the stem cell program, we created this channel. It's called UC Stem Cell Channel. And we have several episodes that talks about the different applications of stem cells. It runs in 3 cable TV channels, so you might end up seeing on TV as well. But on the internet or YouTube, you can see all the episodes. We just passed 1 million of views for these episodes. So we're very excited that the response has been amazing. And the idea is, again, to explain very lay terms the excitement about all these new technology. So I'll finish here by thanking the families, the founding that we have. These are members of my lab. Full disclosure, I'm a co-founder of Tismo, a company that uses brain organoids to study autism. And so happy to take any comments and questions. Thank you. Yeah, so the question is feedback loop. That's exactly what we are closing this feedback loop. So the organoids, we capture the information from the organoids, we send to the robot. There is a command in between that we tell the robot what to do. The robot explores the environments. As it does, it feeds the organoid, changes how the organoids process the information, and sends a second command to the robot. Exactly, that's the goal. Yeah, yeah. Absolutely. Yeah, we are learning how the brain does, so it can apply. Yeah.
Your son, how old is your son?
13.
13. Have you had any improvement in trying to use any CBD products, or has anything helped him?
Yeah, no, we try everything. We try different drugs. I mean, the seizure seems like intractable right now. Yeah, yeah, and so far he's a non-responsive individual. Yeah, so that has been like a major problem. So we end up from time to time on the ER. I'm just back from the hospital, by the way. Yeah, so it happens all the time. And we're not the only ones. We get to know many families where, I mean, seizure is being like a major roadblock. Yeah, so I have his mini brains. We are trying to figure out exactly what's the problem and how to fix it. the question is what kind of cell types or differentiation we have in the organoid. So we start with a very homogeneous population of cells. These are the pluripotent stem cells. And over time, because of the different factors that we use to guide the cells, we transform these pluripotent stem cells into neuroprogenitor cells. These neuroprogenitor cells will form intermediated progenitors that will give rise to different cell types in the brain, glutamatergic neurons, different cortical layers of glutamatergic neurons, GABAergic, the inhibitory neurons, and glia cells, mostly astrocytes that helps on the synapse formation. We can add other cell types. We can add microglia. Microglia are the immune cells in the brain, so you can create them in parallel and infuse them inside these brain organoids. And we are now trying to add endothelial cells to form vases, to fully vascularize these organoids. So these are all coming. Yeah. Yeah. I think if I can summarize like in two questions. One is there any advantage to use the organic networks? And will the artificial intelligence ever reach a level of the human brain? This is debatable. Many people don't think that this will happen. The level of computational power that you need to mimic, even like small things that the human brain does, it's probably impossible to get with the current networks. Some people might disagree. I think it's going to be very hard to model using the algorithms that we currently have. So that's why I think there is an advantage of the organic networks. But there is a major roadblock, which is how it does. And it's all about how the neurons are connected to each other, how they respond to input and output. So this is the project with Microsoft. We are really trying to understand how this would be done. So it's going to be like we hope that's going to be like something similar to the next, like quantum computing, augmenting the power of these networks to a level never achieved before. Yeah. Rina?
Do the mini brains age and do they die?
Yeah, so great question. The mini brains do age. We can see the progress of aging over several months, if not years. We were able to keep them alive for, I think, a maximum of 3 years. And we only stopped because, I mean, nobody wanted to keep growing them anymore. We see that after like a year or so, the center of the organoid, because it's not vascularized, it starts to become necrotic. So the surrounding cells is still alive, and I think you can keep them alive as far as you want, but the center becomes necrotic. So that's why they stop growing. Yeah.
And how do you nourish them?
So
Have you varied the nutrients?
yeah. So we nourish by growing them in suspension in a solution that contains everything that they need: amino acid, nutrients, glucose. So everything that to make these neurons mature and be happy. But you touch a very important point. If we change the way they grow or the nutrients, can we make them mature even further? I think there is still room for improvement, and we don't fully understand. Most of the advances that we have are empirically. Someone tries something and figure out, oh, it's much better. I think we can push the limits. Yeah. I'll start in the middle here. Yeah. So the ethical issues. What are the ethical problems related to this research? And again, I mean, we have no precedent, so we are learning along the way. And just so you all know, I mean, every research that we do in research institutions such as UC or here, it is overseen by an ethical committee. So they keep checking on the status of the research. But what should we do? So last year, September, I invited 3 philosophers of the mind to discuss brain organoids and consciousness. By the way, all the videos are in here. You can watch each one of them discuss their perceptions of these brain organoids. It boils down to 2 things. One is nobody agrees on what consciousness is. So if we don't agree on what it is, it's hard to define. But we agree that there are a couple of experiments that you can do to at least get some initial evidence if these organoids will ever become self-aware. One of these experiments is, for example, if you take anesthetics under anesthesia, your brain changes or silence those brain waves and you become unconscious. So what you're doing, we are adding anesthetics to these organoids while recording from them. Will they lose their oscillations as well? So that might be an indication that they might be conscious under certain circumstances. But let's suppose that they are fully conscious. That's where the problem is. If they are fully conscious, we need to give them a moral status. What would be a moral status for a brain organoid that has a level of activity that would be, let's say, a baby newborn, right? I mean, is it fully conscious? It's not. Is it a person? It's not. I mean, there's lots of debates, and I feel that the organoids would be in the same situation. And we have ways to deal with that. For example, people who work with animal models, animals are fully conscious. You work with mouse models, and they are fully conscious. But what you do if you find a mouse in your house? Right? So I think we have to agree on what would be a set of humanized ways to grow and terminate these organoids for research. And there is the scientists who have to get together with the community members of the society and decide on a moratorium, how far this research can go, how do we grow them, how do we kill them, how many we grow them to answer specific questions. So these are what will happen in the next years or so. I think she was first.
Can anything be done for someone who has strokes?
Yeah, so the question is, can these brain organoids be useful for a stroke or any other brain damage? And the answer is yes, we think that we can do it. So one of the applications is on regenerative medicine, where we can tailor the organoids to specific brain regions to transplant into brains of people who have brain damage. The good news is that we can use your own cells, because I can reprogram your cells, make the mini brain or the brain region that are missing on someone with a brain damage, and replace that. So that's what we think it can have.
I've had 2 bad strokes, and doctors, I've never had any of that.
No, this is just happening in a very clinical research way. It's not even get to that point yet. Yeah, it's very early stages.
2 more questions.
Okay.
You mentioned that there's communication between the robot and the organoids, right?
Yeah.
Can they be made wireless, particularly between the robot and the organoid?
Oh, it's all wireless.
It is all wireless.
Yeah. For Parkinson, there are 2 clinical trials, one here in US and the other one is in Japan. They are not using the organoids because the organoid technology is just like very recent, but they are using dopaminergic neurons produced from pluripotent stem cells. And so, I mean, I think both groups have high hopes as these will be an effective treatment. Yeah.
So we want to thank Dr. Muotri very much.
Thank you.